Yay. All right. We are live. Welcome everybody who is joining us for season 3 of Future of Foraging seminar series. My name is Hannah Habakian, and I'm one of the co-organizers of this seminar. And today I'm joined by Laura, who has been here since the beginning, and Barbara Webb, who will be our keynote speaker today, and then as well as 2 of our 4 new co-organizers, because we've been expanding the team. So today we have Shivam Shitnis and Mai Morimoto joining us, and also new on the team is Emma Scully and Ibo Tashtekin. And then from previous years, we still have Dennis Goldschmidt and Rory Bedford. So if you've joined us before, you know that for the very first episode, we have a keynote speaker who gives sort of a broader perspective on some aspect of foraging. And today our keynote speaker is Professor Barbara Webb. So Barbara studied psychology at the University of Sydney before pursuing a PhD in artificial intelligence from the University of Edinburgh. And then she held lectureships at the Universities of Edinburgh, Nottingham, and Stirling before returning to Edinburgh, where she became a full professor of biorobotics. And her lab's work focuses on how insects with their relatively small and complex nervous systems solve sort of real-world problems. And she focuses on exploring sensory motor transformations in the context of both more reflexive behaviors like phonotaxis in cricket and more complex navigation like you see in ants. And what I think is really special about Barbara's work is that although there is a focus on building computational models of brain function, her lab combines this with behavioral studies, and they're often carried out in the field in collaboration with experimental scientists. And I think this sort of connection really helped her identify what are like the sort of real-world problems insects might face. And her lab also tests proposed models or algorithms by not only simulation, but also implementing them on robots and then testing them outdoors. So I think this approach has been really successful. In many cases, it has inspired neuroscientists like me to follow up with looking for sort of neural implementations of these proposed algorithms. And today's talk by Barbara will be Central Place Foraging, How Insects Anchor Spatial Information. I'm very excited to hear this talk. So it's going to be, just briefly, the format, be a roughly 45-minute talk. And if you have sort of short clarifying questions, please put them in this chat, because Barbara can see that and can respond to that. If you have longer questions that are better suited for the panel discussion, we'll have 30 minutes or so after the talk to address that. And please put those in the Ask a Question box below. All right. So I think we're ready to go.
Great. Well, thank you very much for the invitation. I'll share my screen and hopefully it will all go smoothly. Mm-hmm. And yeah, as Hannah said, I'm actually quite happy to take clarifying questions as we go along, because often it's talking online, you feel like you're speaking to nobody for a long time. So it kind of be nice to know there's a bit of interaction as we go. So I'm quite happy to take questions, and I'll try and be watching the chat for those. So as Hannah said, my group does insect robotics, and although I'm not going to talk a lot about robots today, I thought it might be useful to just kind of give the background that I'm coming from, which is a biorobotic background, where what we mean by that is that we take a very mechanistic approach to trying to explain biological systems. So to understand a biological system, we really should try to build the mechanism and embody our hypotheses as actual machines. And so when we say machines, we don't just mean simulations, we mean literal physical robots a lot of the time. Although we do computer simulations as well. And just, some of the things we've done is to build models of cricket sound localization, multimodal models of escape behavior, models of walking behavior, and even some soft robot models of maggot behavior. But a lot of the work we did, was really fairly simple sensory motor control. And where we've been going lately and being really interested in more recent years is more complex behaviors and, in a sense, behaviors that happen, in the real world, and behaviors obviously that are relevant to this particular seminar series, which is foraging behaviors. And we're focused on central place foraging insects. And this is an example of an ant. This lovely slowed down video of an ant from Antoine Wistrach shows a desert ant that's trying to find its way home in real field conditions. And we know that they forage to bring food back to the nest. And for these species of ants and for a number of other related ant species, they don't use chemical trails to do this, but each ant is navigating individually back to and from the nest. And this navigation is absolutely crucial to their own survival because it's extremely hot in the environment that they're operating in. And it's also obviously important for the colony's survival that they navigate efficiently. They've been shown to do this over very large distances, up to 1 kilometer away from their nest, which is often a tiny hole in the ground, and in complex cluttered terrain, as in this example. And as I'll be describing, there's a couple of key mechanisms that they use, which is path integration and visual memory. So we do field studies of these ants, and this is one from a while ago now, but we've only recently really analyzed the results. So this was work done again in collaboration with Antoine Wistrach and also with Mike Mangan, who is a postdoc in my group at the time, where we were trying to follow ants through their entire foraging careers with differential GPS and then also a downward facing camera. So I'm actually going to focus on the camera data. So this is effectively, although this looks like quite a complex set of equipment, in fact, we can now replace this literally with just one camera and following the ant as it moves around, where we don't constrain where it goes at all, we just follow it. So what this looks like when we were doing the tracking in our ant field in Seville is a bit like this. So we're following ants around. We see we control when they come out of the nest. We have them marked so we know which ant we're following. And then we try to follow a particular ant from the very first time it comes out and then each subsequent time it comes out and to see what it does. And what we have been kind of developing is this fantastic tracking system by Benjamin Risser. So this is actually this is the downward facing view of the high resolution camera. And you can see we're actually able to track the ant, even though it's this cluttered background, that the camera is moving. We can still, and sometimes the ant is even getting occluded under things. We can keep a very good record of where the ant is going. We also, in this particular context, were also getting information about the environment. So also taking LiDAR scans of the environment to try and be able to reconstruct the actual 3D world and the ant is in. And then we could try and create this kind of ant eye point of view where there's this low resolution, like the ommatidia of the ant, and also this pattern in the sky, which we know that the ants can see corresponding to polarization. So just to go back to the tracking system, so this is the kind of global results. And Benjamin, who's now at the University of Munster, has been working with his PhD student, Lars Hulk, to actually not just recreate the tracks, but to then map the tracks back onto the ground surface. So this is actually the tracks of, I think, 14 different ants, all the tracks of these 14 ants from the nest. And each of them is going, as you can see, in a different direction. They're each foraging individually. And this is roughly an 8-meter circle, but some of them are going beyond that. Most of the time we tried to give them food at 8 meters and let them come back again, but otherwise we didn't constrain where they went. And so this shows all the ants, mapped all their trails from following them with the camera, mapped onto the ground surface. And then we can actually zoom in and we have super resolution detail of what the ants are doing. So we can see the little wiggles in their trails, for example. We can see what they're going through, what the ground surface is like. and really start to analyze in detail what's going on in some of these tracks. Okay, so let me talk in slightly more global terms, but about this work. So as I said, we know that ants can do path integration. So basically, this is a naive ant that's coming out of the nest for the very first time. It's taking an outward route, and here the kind of thickness of the line shows the angular velocity effectively. So when it's thick, it means it's stopping and turning around and looking. So it's exploring out, and eventually it finds some food. And when it gets some food, what it does is it doesn't take the same route back that it took out, but rather it takes this fairly direct route back to the nest. And then indeed, once it comes out of the nest again, it doesn't follow the route that it took before, but it goes back towards where the food was and then comes home again. So once the ant has actually done these 2 trips, we actually then try and force it not to use the path integration memory, but to also use visual memory. So here we now pick it up from the nest. So it's done this twice. So it's been out in the world twice in its life. It's been found some food, come home, gone back to the food, come home. That's all its entire experience of the world so far. We pick it up and we put it back at the release point. So back at the food position, when we've taken it from the nest, so it has no path integration. And what we see is that it does a very quick exploration and then is able to follow the route back home, the visual route back home. And as I showed you before, this is in this quite cluttered environment. So it can't see the nest from where it starts, but it's actually already learned visually to follow the route, because you can see, for example, that it goes faster when it's on exactly the same route as it was on before. If it comes off that route, it slows down and starts to look around. And then as soon as it gets back onto this same visual route, it follows it again. So briefly, you can use any kind of camera, or I've actually used this tracking system with a mobile phone camera. The main thing that helps a little bit is to have some kind of gimbal that stabilizes it parallel with the ground, and you keep it roughly at the same height, but even some height variation can be dealt with. So where we're aiming towards here is that it's really very, very simple to collect the data, and then we can apply this method. And we have a paper coming out soon about the tracker. So do get in touch with me if you want to hear more about that. So what I'm going to talk about, though, from now on is really, what more about these 2 mechanisms, the visual memory and the path integration mechanisms. And then hopefully I'm going to go fairly quickly over both of those, and then try and talk about how they get combined. So let's start with visual memory. What's the problem? Basically, this is what the world looks like potentially to an ant. So this is, again, we've taken kind of pictures of the ants from the ant eye point of view along one of these routes, and then we've kind of recreated what it might be seeing and then done it in low resolution as we think the ant eye should be. So what the ant has seen is like the lower one of these videos, and it's seen that twice. And after that, it's able to follow that route home again. So it really seems quite surprising that its memory would allow it to follow this route through a bunch of grass after just 1 or 2 experiences. We think that this visual memory is encoded in the mushroom body, partly because it has to involve some kind of pattern recognition. And we know that the mushroom body in the insect is important for olfactory pattern recognition. And we kind of exploit this idea that was first suggested by the Sussex group, Bart Baddeley and the Sussex group, that what the ant needs to do is not necessarily recognize exactly where it is. It just needs to recognize, does the direction it's going in at the moment look familiar? So this graph is trying to illustrate that. If you imagine that the ant, as it goes along the route, let's show that, stores a little kind of snapshot of what it sees every few centimeters along the route. So that's kind of going from 0 to 800 centimeters here. And And then when it's trying to go along the route again, if it is on the route and it looks to the left and to the right, so that's the heading angle, if it's facing in the same direction as it was when it took the picture, then it should have a very small image difference. So that's what's sort of shown on the z-axis here. And as you go away from that, as it gets further and further from the route, the image difference increases. So essentially what the animal has to do is just follow this valley of familiarity. And basically, you have to associate the views you experience as you progress towards the food or towards the nest. And then to do this association, we think it's happening, as I said, in the mushroom body, because it's an area of the insect brain that's specialized for olfactory pattern learning. And for ants and for other insects that do this kind of visual navigation, there's extensive visual input. And we think the circuitry can perform the function. So this is a kind of a complex picture, which I'm going to go over quickly, but happy to come back to later on if people are interested. But essentially, what we suggest is that it's quite a simple memory mechanism in a way. So the input either originally, so I'm trying to get my pointer to come, but it keeps disappearing. So the input is projection neurons, either from olfactory input or here from visual input. This gets projected into this set of cells called Kenyan cells, and it's a high dimensional projection. So we go from a lower dimension to a higher dimension because there's a large number of these cells, and to a sparse encoding. And then essentially, what you do is to associate the sparse code for anything that you've seen gets associated with which direction you're going. So here, either you're going home or you're going towards the food, and you make an association. And then if you see another sparse code, you associate that, and so on. So because it's a sparse code, you can actually associate many different images in this way. So I think that's a good point about, does it require features in the environment versus pattern matching? So in this model, it's completely pattern matching. We don't do any kind of feature extraction at all. We literally just take the grayscale values and map them to the projection neurons and map that into the Kenyan cells, and then we just learn those. So it's completely a global pattern matching. And probably the insect is doing some more visual processing before things get to the mushroom body, but we didn't want to make any assumptions about the visual processing. So we just did it as this kind of really raw data. And it turns out this works surprisingly well and allows you to make this decision, as I said, am I on the route or am I am I facing off the route? Which direction do I need to turn to be facing the way I was facing originally? And that actually gets you along the route quite well. And it also, even if you're not on the route itself, you can kind of extend this method to do homing if you learn things while you're facing towards the route. So this is what I call view memory, as a kind of more general thing than just route memory. So we have the same mushroom body model, but now we have learning walks or flights. So I think you might have had Paula Fleischman talk to you about some of the learning walks in ants before. So this is data from ants where they come out from their nest, and as they leave their nest, they keep turning around and looking back at their nest. And the assumption is that they're actually learning the view of the nest from different directions. Okay. And again, work from the Sussex group, this time from Alex Dewar, showed that if you put this into the model, if you basically say, okay, I've learned what the nest looks like from these 4 different places around the nest, and now you try and get home from all these other places around the nest, using this simple kind of turn and look and comparison, where do I get the best match between what I see now and what I've seen before? You actually get very good directed information about how to get home from all sorts of directions around the nest that you haven't been before. Mm-hmm. So this seems to be quite efficient and good way to get home. As I said, it's not the only way that the ants get home. They also get home by path integration. And I wanted to just highlight this particular paper by Bickerstaff and Chung, which I think very nicely explains some things about path integration that people often forget. So essentially what you need to do in path integration is to integrate your velocity along a journey. So if you do a complicated outward path, you integrate your speed and direction all the way along that path, and you can do that. That will produce a home vector that points back to your original position. But people often kind of discuss or assume, what frame of reference or coordinate system that could be done in. So it could be a geocentric coordinate system where you're doing it based on your home location is the origin of your axes, or it could be an egocentric system where you yourself are the origin of your axes, and it could be Cartesian coordinates, it could be polar coordinates. And the kind of natural assumption initially when you think of path integration is that it should be egocentric polar coordinates, that basically a home vector kind of implies it's wherever I am now, I know how far I need to turn, which is the theta of the polar coordinates, and I know how far I need to run, which is the r of the polar coordinates, and that would take me home. But as they really nicely point out in this paper, there's good reason why that's not the best solution, in fact. So roughly speaking, if you have polar coordinates to encode your location, then when you're getting close to your location, the change in theta goes to infinity. So basically, you're having to update your r and your theta as you move around, and these are the update equations. But that change suddenly starts to get really fast. And basically, as you cross over your home position, your theta is changing at infinity, which is pretty hard to imagine how you can have something, how you can encode that. Also, for egocentric Cartesian coordinates, that doesn't work terribly well either, because if you imagine if you rotate on the spot, then your x, and you're keeping a track of where your home is relative to you as you rotate on the spot, then your x-axis and your y-axis are changing very rapidly and very continuously. So your measure of x and y have to change very fast, just as you rotate on the spot, when in fact, where you are in relation to home is not really changing, just your angle to it. And then for both the polar coordinates and for both egocentric coordinates, you actually have to use the current state of the vector as part of the update equation. Whereas for the geocentric Cartesian, you just need to know your own movement, and then you can update the equations. So they conclude from this paper, and I think it's quite a convincing argument, and I really encourage people to read it if you haven't seen the paper, that really any sensible way to encode your path integration should be geocentric and Cartesian, okay? And the only disadvantage there is that you have to transform the coordinates somehow to control how you get home from there. Okay, so this will become relevant when I now talk about how we think this is encoded in the insect brain, where we think path integration is encoded in the central complex of the insect brain. And again, I'm going to go over this model quite quickly. I've talked about it in a lot of detail in a lot of other places, but I want to try and get to the kind of combination of the two things. But the essential message is that we know that the right information goes into this neural network. So basically, it gets input from the sky compass and from optic flow. So that's direction and speed. And therefore, it could integrate those two things to do path integration. So it gets input from the sky polarization. And I'm just, again, going through this quickly. So you can measure, responses to polarizers in this central complex. You can measure responses to optic flow in different directions. And both those bits of information are there and come together in this circuit. So this is the model that we've been working on, and it's not, even though it looks like a beautiful, tidy model, it's actually based on real topology from neuro-anatomical connectivity. There are some details, obviously, that are left out, but the essential structure is actually the real structure of this circuit in the insect. And what it encodes, the inner circle here is basically the current heading of the insect relative, using its sky compass. The middle ring are these neurons that get input both from the compass and from speed neurons, from optic flow, as I've shown here. And then that forms, what that will do is form a memory as you move of where you are. And then the outer ring does the steering. And I think my next slide hopefully will explain a little bit how that is one way to explain how that works. So you can imagine essentially as you move in different directions, you're integrating your speed relative to the direction you're moving. And that means what you end up with is a kind of population code across this set of neurons that encodes, so this is your actual home vector, and it's encoded as the activity in this set of neurons. And this is actually a geocentric Cartesian encoding. It's a redundant Cartesian encoding because you have more axes than you actually need, but it still is a Cartesian encoding, and it's geocentric. And then what happens in this circuit is that the connectivity between the middle ring and the outer ring means that it's as though you're kind of rotating this memory 45 degrees to the left or 45 degrees to the right, because you have a columnar shift in the circuit. And you're comparing that to your current heading. And if you basically take the difference here of this, your current heading and your desired heading, your memory of where your home is, so you just subtract these vectors, you can see this is what you get, that you get much bigger, there's a much bigger difference between this one than there is between this and this. There's a bigger difference than between this and this. So this tells you which way, if you just then sum up the overall difference, that tells you whether you should turn left or right to get a better match. So you want to get a better match. You want your current direction to match your home direction, and that you can use that directly to steer home. So let me just show a little animation of this happening. So this is this circuit in operation. We have a little simulated insect going out from its nest, and its current heading is encoded in this middle ring. As it goes further and further away, it's summing up more and more in this outer ring in the opposite direction, which forms the memory pointing back home. And then when we allow it to use the steering circuit, it steers itself back home by comparing the current heading and the memory. And then when it gets near to home, the memory zeroes out. So then if it overshoots, it will just keep coming back again and do a little search pattern, just emerges around the nest. Okay. So I know that was quite quick, but as I said, I wanted to get on to some of the kind of really interesting further implications of this, particularly for foraging and for spatial representation. So we've just talked about path integration, and then earlier I mentioned this vector memory, the fact that if the animal gets home after finding the food, it then goes straight back out to the food again. So that's the blue path here. So what we actually found that we can make a very simple hypothetical addition to our circuit that I just showed you that will do this vector memory. So this is essentially what it looks like, is we just say, when you find the food, you should just store the current path integration state. So here we're just storing it as kind of weights from this vector memory, weights onto these 16 values. And then that basically acts as a kind of inhibition for the steering. So what that means is that when you've got back to the nest, so your path integration has got to 0, you apply this inhibition, and that means your circuit is now going to try and get you back to 0 again, but now it's trying to zero your vector memory relative to your path integration relative to your vector memory. So it's trying to make this difference 0. So that means it will move you to the place where your vector memory is equal to your path integration, or vice versa. Your path integration is equal to your vector memory. In other words, it will take you back to where you started. So let me just show you that again, maybe here, and it will make it clearer. So here we imagine that the animal has gone out from home, it's found food, it's stored a vector memory at the food, and then it's gone home. And when it gets home, it now inhibits its central complex with this vector memory. And that's as though kind of in its imagination, although it's actually at home, its central complex tells it it's over here. So now it tries to go home again. And that means it moves in such a way that what its path integration tells it balances against the vector memory. And because it actually started at home, that means it's actually moving back to this place. But then it couldn't, if it wants to go home again, it can just remove this inhibition, and then its path integration will be correctly at this place and will get it home. But more interestingly, what if it gets to this food place and there is no food? And it doesn't want to go home, it wants to find some other food. Well, if it has some other vector memory that it's stored before, it could basically stop inhibiting with the first vector memory and now inhibit with the second vector memory. So that means it's actually got to here, it's now doing this inhibition, and it's trying to do something that means that its path integration and the inhibition cancel out, which is equivalent to trying to go home. So that's the path it would go. But this, as I said, this is kind of its imagined position. It's actually starting from the food position. And so now what it's going to do while it's trying to take this motion is that it actually is going to take this shortcut to the second food position. So this can actually explain how the animal could do a novel shortcut by effectively vector addition. And we've obviously done simulations to show that this does actually work nicely in practice. And then we can actually extend this to the situation which is not seen in ants, but is seen in bees, called trap lining, where bees are able, if there's multiple food sources, they'll actually go round several different food sources before they come back to the nest. And they've been observed to gradually develop more and more efficient routes around them. So initially, as they gradually discover different food sources, they'll add them into their loop. But as they find these food sources and use them more often, their routes get more and more efficient. So they either go round one way or the other way. This has been compared to solving the traveling salesman problem. It's not quite a solution to that, but it's a good optimization of the route. So again, for using the circuit that I just showed you, we just added in a fairly simple addition. So now, again, as you explore, you store a vector memory for each food source you find. And then when you're leaving a food source, you cycle through all your existing vector memories and try and pick whichever one is closest. And you can establish that very easily by just basically taking the difference between your path integration and your vector memory, which is what's being calculated anyway. So you just say whichever one is this, either is the smallest amplitude or falls below some threshold. That's the vector memory I'm going to use. And then this is our simulation of this gradual discovery of each of the locations, and then a gradually more efficient route. And then eventually we find the most efficient route around. And this is just sort of showing you the kind of statistics of that the length of the route taken drops as we acquire these different things. So we can actually explain this kind of efficient foraging behavior as well, using the same circuit. And then what's very interesting, nice recent thing, so this was a completely hypothetical idea that this is the vector memory could be stored in this way. More recently, Stanley Heinze has suggested that there's actually a specific neuron that is found in the anatomy of bees and not in other insects, not in flies, for example, that seems to have exactly the requirements to fit this, that it basically it's a neuron that extends and has synapses across all the fan-shaped body, making all the connections that we would want. And then there's multiple of these neurons which could correspond to these multiple vector memories that we've suggested. Okay, I have a few minutes left, so let me try and talk a little bit about how the vectors and the views interact. So far, nothing I've said suggests that the animal needs to explicitly associate vectors with views. So you could imagine that it could, if it knows what the view looks like from a place and it has a path integration vector to that place in a vector memory, maybe it could associate those two things together. But the evidence for that is actually not so clear so far. However, the vectors and views definitely have to interact. So for a start, you need to use the vector information to learn the views in the first place. So this is now actually bee learning flights. And the idea is, if you're going to use this method of having views looking towards the nest as a kind of way to get home when your path integration fails, then you need to know where the nest is when you're first learning them. So you need to know where to look back towards the nest to be able to learn the views in the first place. Or similarly, to do the route following, the very first time you follow the route, the only way you can do it is by path integration. So you need the path integration to learn the views, to learn which is a good view or not. A second idea is that the two things can maybe be combined at output stage. So this is basically the idea that if you have both path integration and a view, how do you combine them? Okay, and normally, they should both point the same way, and so that's fine. But there are several interesting studies that have tried to put these two things in conflict. So this is an example where the ants had learned this was the usual food position and the home position. So they'd learned to go this way. And now they're being released from different places where they can kind of see the views. So that would orient them towards the nest this way. But they also have their path integration, which should tell them to go that way. And so what you can see is that they seem to take this kind of compromise position between the two. And we were interested to actually see, could they actually be doing this compromise between the two in an optimal way? Could they be doing optimal cue combination, i.e. weighting each of the cues relative to the uncertainty of the cues? So that's kind of what's expressed here, is you have the uncertainty of the two cues, and you can have a relative weighting. That means the more certain cue gets a higher weighting. So we actually did a study to try and look at this, and we could exploit here the fact that we actually can calculate how the uncertainty should increase with distance in path integration. And again, I'm not going to go through this in detail, but the basic idea is that the variance of your directional information from path integration actually decreases the further you go. And that's basically, you can work it out from the geometry, that it's basically proportional to 1 over d. So even though your positional uncertainty, as you can see, you accumulate uncertainty in your path integration, but your directional uncertainty gets less the further you go by this proportional amount. So that tells us how much ants should weight the path integration versus their view, depending on how far they've gone. And in this experiment, we picked them up at different distances from the nest. So their path integration was 0 or 1 or 3 or 7 meters. And we put them down somewhere where they had a conflict between what their path integration should tell them to do and, put them down here, where their path integration should tell them to run downwards, and their familiar view should tell them to run this way towards the nest. So there's about 120 degrees conflict between them. And what we found, or we put them somewhere unfamiliar where they can only rely on their path integration. And what we found indeed was, if the view is unfamiliar, if they have 0 path integration, they have no idea where to go, but otherwise they follow their path integration. But if they can see the view, if they have no path integration information, they go to the view. But as the path integration length increases, their weighting, they go somewhere in between, and the weighting of the path integration increases. And indeed, it turns out it increases very nicely with the prediction of optimality. So this is kind of showing all the data, and the red line shows what we would predict from optimal integration based on this estimate of how the uncertainty of path integration decreases with distance. Okay, so now I think this is my final little section that I'm going to talk about. There also appears to be directional information transfer between views and vectors. And this came from an observation that ants I just told you that ants, follow the view by turning so that they're matching. They get a match in the image between what they see and what they do. But ants often go backwards. So this is an ant dragging a dead cricket backwards, and it's going straight towards its nest, even though it's going backwards here. So what we wondered was, okay, how is it doing this? Is it actually able to match the view from any direction, or is it somehow getting some vector information on, for example, as you can see there, these occasional times it seems to look around. So this is a more controlled experiment that we did to try and understand this. So essentially we trained ants to go around this little slalom, and then we basically wanted to see which way they went. So we gave them a big piece of cookie, so they had to go backwards, and we would pick them up from, let me see if I can try and get this clearer. So we'd pick them up from here, so their home vector would point in this direction, and then we'd place them over here, so their view familiarity would point in this direction. This is the route that they should follow. And we found indeed if they're going forward and can look, they'd follow that, and if they're going backwards, they would follow the home vector. But what some of the ants would do is drop their food and have a peek at which direction to go. So this is an example. So this is an ant that's following its vector backwards, and then it drops the food, looks around, and then when it comes back to the food, it takes it in the direction that the view would tell it should take. And this is another example. Where they just need one quick look at the view and then they know which direction to go. I think I'll leave that one. Yeah, okay. So this is the kind of summary that basically the direction they were going before they took a peek and the direction they're going after they took a peek. It looks like they can actually transfer the information, so they can align themselves with the view, and then somehow transform that into a vector that they can then, a vector information that they can then follow relative to the sky. And yeah, okay. So in the interest of time, I'll go to my conclusions. So what I've tried to show you here is that we're trying to connect these kind of insect navigation field studies to neural mechanisms. And obviously it's quite hard to do this by direct brain recording or manipulation. It's hard to do that in the field, and it's also hard to get animals to do real foraging tasks in the lab. So modeling here provides a link between what we know about the neural mechanisms and what we can see in the field studies. And at the very least can demonstrate the plausibility of some of these algorithms to be implemented in the insect brain. And we also gain complementary insight into the circuit function. So we've kind of suggested that the mushroom body architecture is a kind of general purpose multimodal associative net, and that the central complex architecture is actually a general circuit for steering by comparing a remembered goal to a current heading. And I haven't really shown you this in this talk, but we also, I think robotics kind of provides a framework here, both for evaluating our work, but also just for thinking about the problem. So we always start, when we look at these problems, we always start by saying, well, how would I do that on a robot? How could a robot solve this problem? And then we think about what in the insect brain might set constraints on those solutions. And that's very important in how we kind of come up with some of these ideas. And then I'll just thank my group members. The dark ones are previous group members, and the current group members, and my collaborators. And thank you for listening. And I'm very happy to have further discussion.
Thank you very much. This was a really wonderful talk. I'm going to invite the other panel members. Sorry, I have to remember where to do this. Here. All right. Just 1 second. And everybody else, please, ask questions. if you have something you'd like us to address in the next few minutes. All right.
Okay, I can see there's questions there, but I'll let you.
All right. Yeah, we can probably. Sorry, I just have to find people in this list. And I don't. All right. Okay. Let's see. I've invited Rory as well. And Shiva. So anyway, I hope the rest is going to join us in a second. Let me see.
Should we just start asking some questions that's been posted already?
Yeah, exactly. So maybe I think the pheromone question is a good one to start. So
sure. Yeah. Okay. So I think for the ants that we're studying, the desert ants, I think it is safe to say that they don't use pheromones to mark trails. So they do still use smell, and people have shown that they will use, if there's odors, like if you put odors along the route, that becomes part of their memory. So if you then change how the odors are distributed along the route, they notice that something has changed. But that's not a pheromone trail, and it's basically because they have to move really fast in the desert, and because the ground is really hot, so pheromones burn off, and also food is very sparse, so in general they're not all going back to the same place, although if there is one food source they will all start to go to it. So they do have some mechanisms for other maybe communicating where the food is, although we're not sure what that is for the ants at least. But I think it's safe to say that they're not using pheromone markings for the behaviors that I showed. And in particular, we were showing behaviors where it's, a single ant. The other thing to mention there is even ants that do use pheromone trails, people have done tests. If they use a pheromone trail to go to the same place multiple times, at least some of those species will actually learn it visually. And then if you put it in competition, if you give the pheromone trail in competition with the visual surroundings, they will use the visual surroundings because it's actually faster if you've remembered the visual surroundings. So even ants that do use pheromone trails will also use this visual memory mechanism.
Thank you. I think we can sort of group a couple of questions here. Maybe first another one that is basic and sort of on its own is from Rory. I'll just ask it. So is the specific central complex circuit architecture that you mentioned conserved across insects, or do you think networks like these are likely to be more sophisticated in species with an ecological home on which to base path integration computations?
Yeah, that's a good question. And Stanley Hines is really the person who should answer that question. So I'm really just kind of paraphrasing what he might say. So I think the short answer is that they are it seems to be very conserved, really surprisingly conserved across all invertebrates, even not just insects. So that suggests it's not just for path integration, and we think it's involved in steering, any kind of goal-directed steering. On the other hand, there do seem to be some special adaptations and some differences that we observe in insects that do central place foraging and that require path integration. Having said that, there's some evidence now that at least that Drosophila will do path integration over shorter, smaller scales. So they'll path integrate back to the location of somewhere where they found food recently. And I think we're going to find path integration actually exists in a lot more insects than we appreciate. There's one paper, I think, showing it in crickets. There's papers now showing it in some other arthropods. And so, yeah, so I think it may be more widespread than we realize as well. So,
can I ask a quick sort of follow-up? So I guess there is maybe some requirements on the central architecture side, but like even for the sort of more fancy view-based learning, the insects don't require actually a lot of specialization on the sensor side, right? Like all of this can be done with relatively coarse vision. And I mean, I don't know if there's any evidence for developing any specialization basically in the earlier visual system.
Yeah, so I think in our models we can do it with quite coarse vision. The only thing I'd mention there that we've found is that having UV vision might help, because it helps you separate the sky from the ground and to get a very distinct skyline. And we've shown that can actually be a very useful way to learn this kind of global view. So that's maybe one specialization, but otherwise, I don't think. And even the mushroom body, I mean, so what is again different between insects there is the number of neurons in the mushroom body. It goes from like around 2,000 in flies to 200,000 in bees and ants. So again, that probably implies that, but the actual structurally, there's a lot more similar than there is difference. I mean, it depends who you ask. Some people emphasize the differences. I like to emphasize the similarities, I guess.
Yeah, makes sense. Do you want to? I think Laura, you had a sort of question that is sort of related. Do you want to ask?
Sure. Then now I have other questions.
Yeah. You can.
I'm just trying to think which questions. Right. So, I mean, I guess I was also. I mean, okay. I'm kind of interested in the idea of like how flexible the memory encoding is, and I'm wondering about sort of if the environment changes, for example. But also actually now thinking about different temporal scales as well. Like, I mean, what kind of evidence is there for ants, for example, using the same vector memory like across days? Sorry, I'm not an insect person, so I know a little less about these things. But yeah, I'm just kind of, because you mentioned different vector memories, for example, sort of be like having different influences, or and I'm just kind of how those things interact and how they're encoded over to different timescales. That's very
Right.
Yeah.
So in brief, I mean, the memory seems to be quite long lasting relative to the lifespan of these animals, which is short. Okay. For ants, they usually, they forage maybe for a week. But they seem to be able to, that memory seems to last for that. It only lasts over days, they'll come out the next day and they remember where the food is. Yeah. For the question sort of about changing the scenery and so forth. So what people have done is shown that, they do. It needs to be a fairly major change. So sometimes things we think, we'll move this big landmark, and they don't seem to pay much attention to that. But it turns out if you look from the insect's point of view, often those things are not as obvious as you think. So if you're using the whole scene, then those things don't matter. But if you do something like chop down all the vegetation around their nest, then they come out and immediately they start to do these learning walks again. So it's as though they realize, okay, nothing looks familiar. I need to relearn. Yeah. I'm not sure if does that answer. I mean, there's probably lots of questions. And then if we get into bees, it all gets more complicated as well, what they can do. But
I mean, I guess also in the encoding side, and again, as somebody who knows like of the mushroom body, I know of these things, but I'm not in great detail. Like exactly how you could even encode so many different potential like vectors, for example, if there are multiple, like when you were saying, for example, in the sort of multiple place location foraging. And how those things interfere if one is sort of, if they're a little noisy, for example. I like to me, I felt like the way it sort of seems is that it's a very sort of like the fidelity of the representation like seems really high, and able to use this kind of very non-noisy information to be able to direct their paths. Whereas I guess there must be, I'm thinking noisiness internally and externally, so in painting environment, but also in terms of exactly how it's encoded as well.
Right. Yeah. Okay. I'm trying to think how to answer that. I mean, we haven't looked systematically at noise. I mean, we do include noise in our simulations, in some form, just to make sure it's not, all perfectly clean and clear. I think and I think in terms of actually like pinpointing locations, I think, the insects have multiple strategies there as well. So I think, for getting to the nest, you just need to get near enough to the nest. You don't necessarily have to get right to the nest, because then there are other cues, like maybe chemical cues or again, other visual cues that you can use to pinpoint it. So I don't think they're doing pinpoint accuracy with either of these things. Yeah, if that helps. But it's good enough to get them back in the right vicinity. Yeah.
I think we have, I guess, 2 questions that are sort of summed by maybe dealing with uncertainty and also maybe using past experience. So, Shivam, do you want to ask your question?
Sorry, everyone is muted right now, just to make sure everyone's aware. Shivam,
yeah, I can ask your question otherwise. Let me know. I think it doesn't find the microphone because I can't unmute him either. So there's some. Okay. Well, I will be Shivam for a second. So he was asking, does that, and I think that was in relation to the experiment where you showed views and homing vector conflict, mean that insects would know the uncertainty of each estimate to combine them? Is there a plausible circuit mechanism to do this? At least that was my guess of what it was about. And is, I think, related to a question I had, which was whether if you know something about uncertainty, maybe also from experience, could you control this experimentally, could they weigh these cues differently and essentially switch between strategies?
Yeah. So what has been observed is that they, the more they do the root, for example, the more they start to rely on the root memory. So the visual memory certainty seems to increase over time, and they'll start to weigh it more over time. Whether they're, how the uncertainty is kind of encoded, as it were, that's quite an interesting question, and it's quite nice how, in a way, in this central complex circuit, if you imagine, this population code that I showed you, you can, to some extent, you can kind of encode uncertainty in that in a very natural way. So basically, it's the equivalent to the length of the vector, gives you the uncertainty estimate. So you can actually have, if you have 2 different sources of information about which direction you should go, but one of them has a much bigger difference between the activities than the other one, then that would just naturally be a stronger effect. So effectively, it's like adding a longer vector to a shorter vector, and it will have a stronger effect. And you can kind of show that that's, for a circular problem, that's actually the optimal way to do the combination. So it kind of all falls out quite nicely in that circuit. I'm not sure if that answers the question completely.
Yeah, I think now you can hear me, right?
Yeah.
Yes.
Yeah.
Okay.
No. So just a quick follow-up. So in that case, then the uncertainty of the visual memory would be equivalent to, what you get from the mushroom body in that sense? Like how much the mushroom body modulates that vector would just be the uncertainty of the visual memory in some sense?
Yeah, I guess that's what we would argue, is, yeah, exactly that. If you have if your mushroom body is more strongly signaling that this thing is familiar, then how that gets integrated into the central complex for steering would be a stronger effect. And I'm kind of waving my hands a bit at the moment, but we are actually doing some models now on how these things, what the neural system could be that does that combination. That's kind of ongoing work in my group at the moment.
Very cool.
Is to put these 2 things together, in a more systematic way, and following what we know about the neural connections now between the mushroom body and the central complex.
Right. Yeah. Sorry.
Yeah, so I was going to follow up on that and ask about those peaking ants in the end, because that was very sort of an adorable behavior. So what do you think is going on to trigger that peaking behavior? So do you think that the uncertainty has reached a certain level and that?
Yeah, I mean, I think that's really a good guess. I mean, at the moment we don't really know, but I think it's a very interesting question. Exactly what drives them to decide, that they're uncertain. So one thing I think we found in that experiment, and I must admit I don't remember the details. I was going to reread the paper before I talked, and I didn't. But I think what we found is, the shorter their vector, the more likely they are to peak, basically. So they would certainly peak if they had a very, if there were zero vector ants, then they would definitely peak more quickly. And whereas if they had a good long vector, then they wouldn't peak as often. But I don't think we did that systematically. I think that's more just a kind of, an observation from the studies that we did. But yeah, I think it's a good question about, exactly, could we actually find out more systematically that there's something that triggers the uncertain, the level of uncertainty gets to some point at which they decide they're going to peak.
I have a question going in a slightly more foraging, I guess, related thing. But kind of on the point of different memories, etc. Is there any work on associating a memory of a food source with also the type of food source, the value? And is there anything done on sort of selectively, selecting a memory based on your current needs? So I think there is some specialization for sugar versus protein foraging in ants as well.
Yes, yeah. So there definitely seems to be something like that going on. More probably people have done more on that in bees than ants. But so bees, for example, seem to remember for flowers that open at a different time of day, they'll know, this is when I should go and forage on. I should activate this vector memory to forage in that direction rather than another one. There's some interesting stuff with ants where people have done food of different value, and then will they go further to get the food of a better value or not? So I think there probably is something, it's not just a memory that there's food there, but it's a memory of something to do with the value of that food. And interestingly, might be also that they might learn some negative things as well. So they might have memories of somewhere that was bad. And the evidence for that in bees comes from when, so bees will dance on the honeycomb to tell another bee, here's a vector to food. And if a bee is dancing towards a food source where another bee has actually been punished, has experienced something tried to capture it or whatever, the second bee will actually basically bump into the first bee that's dancing, as though to say, well, stop dancing, that's a bad place, don't go there. So that's really interesting that they can both interpret, where the other bee is referring to and have some kind of negative value, in this case, associated with that place instead of a positive value.
Yeah, that's crazy.
And I think the same might happen for views, that they might learn negative views as well as positive views.
I guess, because there is, sort of traplining is nice because it's in some ways very efficient, but it could be that you are that you can take a slightly longer route to avoid something bad, right? And we lost Barbara.
I think.
Am I?
So okay. You're okay, just to let you know. Yeah.
Yeah. I'm not sure what happened. We'll see. It says waiting to reconnect for me, so I'll see if I can invite her back in. Accepted and connecting, it says.
We got too far through this episode without any real tech problems.
I know, this was way too smooth.
At some point, surely.
And then maybe we can have a is there Evans? What else?
Yeah, sorry about that. It's just crowdcasts seem to just randomly did something funny there. I'm sorry about that.
No, it is not your fault. We just said it was very smooth so far. So
spoke too soon. Sorry. So was I in the middle of saying something about? No, I think.
No. I was just rambling and thinking like how basically if you factor in other stuff that could happen during the foraging, I can see how having these different strategies could also help you basically achieve multiple goals at once, right? And the view matching based like route following has some advantages. Basically, you could probably integrate aversive cues in there. There was another question. So I'll from Alison Comrie. And she asks, is there evidence in your models or neural studies for sequential neural activity iterating through potential next or prior foraging sites in ants or bees? Curious to hear about how this might be instantiated as they take and refine shortcuts.
Right. Okay. So
it's a long question.
Yeah. And it feels like there's kind of two things there. So one is, is sequence matter at all? For example, in the roots, I showed, the model that I showed didn't really have any sequence to it. It was just at any point in time, you're just saying, does this look familiar or not without sequence? We have actually done some more recent models where we included some sequential information there, so that we basically had motion inputs, and you're actually learning how the world is changing, which kind of is like a short sequence. So in that sense, and that does work better. So having a bit of sequential information is better for recognizing the route. But I think if I understood the question anyway, that it was going towards was a little bit more, does the foraging bee that's doing a trap lining, does it kind of, think about where it's going to go next?
I think so. Yeah.
And almost, and I guess in our model, effectively, that's sort of what it's doing. It's like running through its memories in its head and then saying, okay, this one, how far is that one? How far is that one? How far is that one? That one's close enough. I'm going to go there. And that's just how we implemented it. Whether there's any evidence that the insects can do that, I don't know. But they certainly seem to find the more efficient route. And I think it's hard to imagine how they could find that more efficient route unless they do some kind of internal comparison of, here are the possible places I could go next and which one am I going to end up at next. But there might be some other mechanistic way to solve that doesn't require them to actually, imagine first where they're going and then go there. I hope that's answering the question. Feel free to clarify if that doesn't quite get at it.
Yeah, I'll keep an eye. And maybe we sort of end on something that you ended your talk on as well, which is a question about robotics. This is from Rory. Please, could you expand a little on your experience of using robots to reproduce and test these algorithms and what you see as the benefits for using them over computational models?
Okay. So we always learn something from when we build it onto the robot and test it in the real world. There's always something we realize we've kind of skimmed over in the simulation and just made some assumption about, what is possible to sense or what might be the type of noise. So for example, in the path integration, we've actually put that now on a flying robot, and we're now getting the noise from wind information, wind noise rather than some other kind of noise. And it turned out to be quite robust to that, which was nice. But the other problem was height, and we hadn't really thought about height at all, but height changes the optic flow quite substantially. And it turns out it's robust to a little bit of height variation, but not to a lot of height variation. And so that means we have to kind of now try and solve the problem, well, how might height be estimated, or how can it be compensated? Or is it the case that bees are trying to fly at a constant height? That's another option. But it's just every time we do it, we put something on a robot, we find something that we've missed. And so after many years of doing it, it's also kind of, as I say, it works a little bit the other way around as well, that just whenever I start to simulate something, I'm still thinking about it in robotic terms. So I'm trying to think, well, it's very tempting to, for example, to simulate a neural circuit where what you simulate is the phenomenon in the neural circuit. So, for example, this bump in the central complex, I mean, it's amazing and it's really interesting. And there's lots of interesting models of it. But in the end, the bump doesn't matter. What matters is the behavior of the animal. It has to be good for something. And you can't really evaluate whether you've reproduced the right properties of the bump unless you're using it to control the behavior, because the properties you're reproducing might be completely pointless, ones that the animal doesn't care about, or there might be completely crucial ones that make the whole system work or not. And I think unless you think about it through from, what's the sensory input to what's the motor output, unless you think about the problem in that way, you can often get misled into what's important to reproduce or to understand. And so that's what I kind of think of as a robotic frame of mind or point of view, is that if I'm thinking, how could I put this on a robot, then I try and think about all these things from the beginning rather than kind of plug them on later to the model. So I hope that answers the question. Yeah.
It made a lot of sense to me. Rory can share it. Here's a follow-up question, I think. Very interesting. So what kind of sensory information can these robots detect, and how does the acuity of these sensors compare to the ants?
Okay, so we've used optic flow on the robots, and that works fine. And we can use low resolution, and we've done the visual recognition process. We've done that with low resolution vision, and that works quite well. We're just currently working on a polarized light sensor to get the sky compass information, and try and estimate, can we get good enough compass information to really do this navigation. So we are trying to reproduce the sensory systems. Yeah.
Well, thank you. We've had we've been going for roughly 30 minutes the discussion. I think I really enjoyed the whole session. So thank you. And unless we have anyone else with a burning question, I would say, well, okay. Well, thanks again, Barbara, for joining us and for this presentation. I think it was really good, very different view from what we've had so far. And,
okay, good.
Yeah.
Thank you.
Thank you. I will.
And we have an episode. Actually, the next one is coming up in just a week. It's soon. Okay. So just a heads up. It's going to be at the same. Wait, it's going to be an hour later. The time change is hard for us. It's an hour. It depends where you are. Okay, just check on our website.
No. Check the website. Yeah.
Yes. And it's going to be on the topic of group foraging or collective foraging.
Yeah. Good. Thank you.
Thank you so much, Barbara.
All right.
Bye.
Bye. Bye.